Disease ecology is, at its heart, a multimodal and multi-layered perspective on the role of disease in the world. Disease ecologists look far beyond the manifest symptoms. They consider the infectious agent, the individual host, the population of hosts, physiological and situational risk factors, the natural and built environment, and the complex ways in which all aspects of the ecology of the disease interact with one another. In the design world, we call this a wicked problem, a scenario or issue in which it seems there are too many factors, too many variables to ever find a permanent solution. There will never be an end to disease, just as there will never be an end to poverty, hunger, or death. Perhaps that sounds cynical. It certainly sounds daunting, especially for those of us who work to reduce hunger and break poverty traps, to cure diseases and prolong life. But I am not cynical, and I am not daunted. As a designer, I face wicked problems every day. I work to solve unsolvable riddles and test untestable hypotheses with too many confounding variables to count. Perhaps that sounds like rocket science, but it isn't. Design is harder. What excites me about disease ecology is how much it mirrors design. Both are highly complex, a system of systems. Both are inherently transdisciplinary and thus require collaborative interventions. And when leveraged to their full potential, both disease ecology and design ecology have the potential to radically improve planetary health. I'm particularly interested in leveraging design to reduce the burden of disease and to improve food security and resilience in the modern urban ecosystem. Because an urban problem is looming. More than 1 million people move into cities every week, and by 2050, more than 75% of the global population will be urbanized. Cities are now the dominant factor in economic and political change, the primary origin of pollution and disease, and the largest consumer of global resources, including food and water. Supply chain security of food and water are fundamental drivers of the growing urban problem. Some of this food is sourced right in our own backyards. Backyard poultry are an important subsistence resource for the poorest communities around the world. But backyard flocks also pose a demonstrable threat to human health when they act as pathways for pathogen transmission. Backyard flocks have also become much more common in the US and other industrialized countries, including in middle and higher income communities where birds may be treated as pets. This growing population and changing landscape of human-animal interaction make the need for better data all the more urgent. Backyard poultry typically have a low biosecurity level. Free access to the outdoors may increase exposure to zoonotic pathogens, and close proximity with owners makes it possible to pass those pathogens on to human hosts. Safety routines and procedures are poor in comparison with commercial biosecurity measures. Backyard poultry are not an insignificant factor in the epidemiological triangle of infectious disease. Worldwide, 80% of flocks are backyard flocks. Infectious diseases are humanity's greatest natural enemy and leading killers worldwide. There's plenty of data on backyard flock transmission of pathogens, such as influenzas and enterics, bird to bird and bird to human, that should inform successful strategies for prevention, control, and elimination of disease. Despite all we know, however, backyard flocks remain a persistent reservoir for infectious diseases around the globe. Diseases like NTS salmonellosis. Non-typhoidal salmonella, or NTS, is a gram-negative bacterium that causes gastroenteritis, bacteremia, and focal infection. NTS presents a significant global health burden. It is responsible for an estimated 155,000 deaths and 93.8 million cases of gastroenteritis each year. An estimated 86% of these cases are thought to be foodborne. NTS is the leading cause of hospitalization, leading cause of death, and second leading cause of illness among 31 major pathogens associated with foodborne illness across multiple countries. In the United States, 85% of all Salmonella enteritis infections are caused by NTS, an estimated 1.2 million cases, more than 23,000 hospitalizations, and 450 deaths each year. Acute diarrhea is the primary clinical presentation of an NTS infection. Diarrheal disease is a leading cause of global illness and death, especially for children under 5. Diarrhea kills more children than malaria, measles, and AIDS combined. Worldwide, diarrhea was the eighth leading cause of death in 2016, the 5th leading cause of death for children under 5 years old. Unsafe food and unsafe water are leading risk factors associated with diarrhea. The most common source of NTS exposure is through ingestion of the Salmonella pathogen. Known NTS transmission pathways include direct and indirect contact with infected animals, feces, and food. Direct contact from person to person or animal to person is a significant pathway of transmission. Animals in close proximity to humans can serve as carriers for NTS, even if they display no symptoms themselves. Another significant pathway is vehicular transfer through contaminated food and water. Meat, dairy, and egg products all transmit NTS, specifically raw milk, raw or undercooked poultry, and raw or undercooked eggs or egg products are common methods of vehicular transfer of NTS. A wide range of produce, sprouts and leafy greens, fruits, and roots also have been associated with outbreaks. An NTS infection begins in the small intestines, where the organism adheres to and invades the intestinal epithelium, causing a strong inflammatory response. Symptoms emerge 12 to 72 hours after ingestion and include diarrhea, fever, and abdominal cramps. In the absence of complications, the disease is typically mild and does not require medical treatment. Most individuals recover in 1 to 7 days, but shedding of salmonella through feces typically continues for days or weeks after symptoms resolve. Up to 5% of cases designated as invasive NTS or INTS involve bacteremia, where bacteria leave the intestines and enter the bloodstream, causing a more serious systemic infection. Hosts who are malnourished or immunocompromised, as well as seniors and children under 5 years old, are at higher risk for serious illness or INTS. It is also possible to develop a focal infection, which requires antibiotic treatment. Diagnosis of NTS requires culture of specimens from the infection site, typically blood or stool, but specimens have been collected from joints, wounds, abscesses, and other sources. Stool samples are most frequently used to diagnose NTS, but in focal or systemic infections, stool cultures are generally negative, so a blood culture is needed for confirmation. Diarrheal symptoms may be treated with oral or IV fluids to avoid dehydration. Antibiotics may be administered to high-risk patients, or when a focal or systemic infection is present, but are contraindicated for uncomplicated cases of gastroenteritis, as they do not shorten the duration of the disease and may prolong shedding of the organism, increasing risk of transmission to other hosts. If antibiotics are prescribed, antimicrobial susceptibility testing should be done to determine which drug may be most effective. Antibiotic resistance is relatively common with NTS. Based on known pathways of transmission, infected individuals at a higher risk of acting as a carrier, such as healthcare workers, daycare workers, and food handlers, may need to be treated with antibiotics and monitored with follow-up stool cultures to confirm eradication of the organism. The single most important factor in the prevention of NTS is avoiding contaminated food and water, so traceback techniques are paramount. Contaminated food can travel for thousands of miles, crossing multiple jurisdictions and serving as a pathway for transmission of the pathogen across a geographically dispersed area. So case reporting is essential in traceback. In the US, passive surveillance of laboratory-reported data is provided through multiple agencies, and active sentinel surveillance of a representative population is provided through FoodNet. Human salmonellosis cases are also reported to PulseNet, the National Laboratory Network for Detecting Foodborne Illness Outbreaks, as part of the Nationally Notifiable Diseases Surveillance System. PulseNet uses standardized molecular serotyping and subtyping techniques to fingerprint bacteria, tracing geographically dispersed outbreaks of the same bacterial isolate back to a common source. Human cases of salmonellosis are reportable but it is not known what portion of infections are actually being diagnosed and reported. Serotyping is critical. It's critical for epidemiological research, surveillance, and intervention. It can assist in differentiating isolates, aiding traceback techniques. Non-typhoidal Salmonella is one of almost 2,600 known serotypes of the genus Salmonella. More than 99% of human salmonellosis cases are caused by subspecies Enterica 1, which includes both typhoidal and non-typhoidal Salmonella. Serotypes within S. enterica subspecies 1 are differentiated by sequencing techniques. Currently, pulsed field gel electrophoresis, PFGE, or whole genome sequencing, and the immunoreactivity of O and H antigens. The identified serotype is then designated by a unique formula and name. Many Salmonella serotypes have broad host specificity, meaning they are generalist pathogens adapted to human and non-human hosts. In total, subspecies 1 includes over 1,500 other serotypes, but the 20 most common serotypes from human specimens account for about 70% of all identified isolates. Salmonella undergoes genomic variation with each new infection. The host's own genetic history, immune response, and application of antimicrobials through medical intervention, all of these can result in adaptation and evolution. Salmonella has a strong incidence of antimicrobial resistance. Exposing the pathogen to antibiotics increases the likelihood of producing more resistant strains. And because antibiotics also tend to prolong shedding, it also increases the likelihood of passing those strains on to others. Salmonella exhibits an amazing ability to survive in the face of adversity. It thrives in wet, shaded environments, can survive in pH 4 to 8+, can grow in temperatures 8 to 45 Celsius, can adapt to low oxygen environments, can survive for extended periods outside of a host, in soil, water, dry matter, dehydrated foods, spices, and candy, for up to 1 year or more. Salmonella is resistant to the negative effects of drying, salting, and smoking, but remains highly sensitive to beta and gamma irradiation. Lessons learned from commercial poultry and egg production environments suggest that environmental factors play a central role in Salmonella transmission. The best prevention measures within the environment seem to involve optimization of sanitation protocols, pest management protocols, and food water supply chain biosecurity. These efforts in relation to overall flock health provide the best protection against the most serious risks from Salmonella infection. Backyard flocks provide food and economic gain to millions worldwide, the potential to reduce malnutrition, break poverty traps, and provide income for advancement. But they also present the clear potential to serve as pathways for zoonotic transmission of pathogens like NTS Salmonella. As the global population becomes more and more urbanized, and as backyard flocks become more and more common, the threat posed by NTS will continue to grow. Diarrheal disease is a leading cause of global illness and death, especially for children under 5. Diarrhea kills more children than malaria, measles, and AIDS combined. NTS specifically is the leading cause of hospitalization, leading cause of death, and second leading cause of illness among 31 major pathogens associated with foodborne illness. Unsafe food and unsafe water are leading risk factors associated with diarrhea. NTS Salmonella presents a wicked problem. Salmonella exhibits an amazing ability to survive in the face of adversity. It exhibits broad host specificity, leverages the immune response to its benefit, has evolved to survive and even thrive in less than desirable conditions, and has a strong capacity for antimicrobial resistance. Disease ecologists and design ecologists may benefit from shared research initiatives to address infectious diseases like NTS Salmonella. Both fields faced wicked problems that are highly complex. Both are inherently transdisciplinary and thus require collaborative interventions. And when leveraged to their full potential, both disease ecology and design ecology have the potential to radically improve planetary health.